Now we will move on to the second session of the day, which is yet another interesting topic on the usage of mitochondrial DNA and Y-SNPs in forensic science and population genetics. The discipline of forensic science necessitates the principle of individuality. No one is similar to another. Deoxyribonucleic acid plays a vital role in forensic science through exonerating the innocent and convicting the guilty. The genetic material in DNA allows the identification of the perpetrator towards individuality and chemical nature. Mitochondrial DNA has enormous potential in forensic genetics, allowing identification of genetic material from degraded samples, dead tissue, and sometimes ancient biological material. Genetic testing can also be performed using mitochondrial DNA coding regions, that is SNPs. Single nucleotide polymorphism have a number of characteristics that make them unique for forensic analysis, allowing the successful analysis of degraded samples. Single nucleotide polymorphism in the non-recombining portion of Y chromosomes, that is Y-SNPs, have important value in anthropology and forensic science because of the characteristics of patrilineal inheritance and genetic stability. To take us through this interesting field of biology, we have in our midst Dr. Arjun Rao, Assistant Professor at Christ University, Bangalore. Dr. Arjun began his academics with a graduation in microbiology and master's in forensic science from Amity University, Noida. Sir was awarded the JRF and SRF grant during his PhD at University of Mysore. Sir's PhD was titled Molecular Phylogeny Among South Indian Population by DNA Fingerprinting. Sir has a rich teaching experience of more than 10 years at Guru Ghasidas Vishwavidyalaya, Bilaspur, Chhattisgarh, and presently at Christ University, Central Campus at Bangalore. Sir has presented close to 15 papers in national and international conferences. Sir is the author of 4 book chapters and more than 25 research articles. Sir has also been a resource person for various seminars and lectures in India. Sir's interests include DNA fingerprinting, population genetics, and genetic markers. When we reached out to Sir for a talk on mitochondrial DNA and Y-SNPs, Sir was gracious to accept the invitation. Thanking Sir for his support, I call upon Sir to address the session.
Yeah, thank you, ma'am. Am I audible?
Yes, sir. You're audible.
Right. So, good afternoon to all of you. So, usage of mitochondrial DNA and Y-SNPs. What we always go through behind DNA or DNA identification, like we always say like 99% DNA is not at all similar, but only 1.01% is similar, which makes us unique and individual. So there are few things which we need to first know before moving further, you can say. Okay, everybody knows that DNA profiling or individual's DNA, how it is different or how it will be identifiable and all. But before, like in forensic perspective or a human population, okay, you can say broader aspect, human population. Especially, we always settle down to the forensic community, which only says that linking a suspect to a crime scene. But beyond that also, there is a world called as population, especially human population. We always run behind the criminals, suspects, and individuals and all these things. But how to make a profile, okay? How to make an individual DNA profile, that makes important. What now in Indian context, it is not running at all. So like European models or American models, when you will see, then they had already established the DNA profiles. Okay, they had already established the DNA profiles, and they are telling that this particular community is having this type of original of markers or this type of nucleotide positions and all. So one way is that how to establish a DNA profile, and the other way is that how to interpret the results, that whether the individuals are similar or whether the individuals are different. So before going to only DNA, or before going to only making a profile, okay, DNA variations, which occurs in the huge population level. Okay, it's not only like a family, or it's not only a smaller community, but it happens in a population. So why I had written forensic and population genetics is because we have two different types of studies perspective to forensic science. The one is always what we search for, that is criminals and suspects, and the another is the population in which we are working for. So there were so many different types of works were already done previously, in which they had prepared different types of catalogs, different types of Y-SNP catalogs, mitochondrial DNA catalogs, YSTRs, mini satellites, microsatellites, and all. But the basic differences is that from the Caucasoid individuals and the African individuals, we majorly differentiate with minimum or maximum 8 different nucleotide differences. So if these particular differences we can identify, then we can say that what is the race of the individual sample, or what is the race of the individual sample, what you had recovered from the crime scene, or from a population perspective also. But this is not the constraints, okay? Not like only forensic science or only population, but there is a vast issues to be underlined here. That is like missing persons individuality, or either in a plane crash or mass disasters. There are so many different types of cases we heard recently. Okay, either it is due to the in Indian context only, or abroad, or some other different areas where the DNA was already defined as a gold standard. Right. There are general basic things are there. Okay. When past 10-15 days, 10-15 years we used to work, then we used to take like 5 ml or minimum 7 ml of blood samples, and then after going for a DNA extraction. But nowadays it's very much simple. Okay, it just you can go for a microliter of biological sample, either it is a blood, saliva, it may be semen, it may be urine stains. But urine stains is specifically that if any male or female had encountered any sexual type of acts with the previous duration, then only you can use the urine samples. Then after the hair samples, hair strands will be there. DNA extraction, there are enormous methods are there. In 3 hours also, now you can extract the DNA. It is very much easily available. But the problem is that when you will be using a kit-based methods, okay, kit-based methods, the DNA can survive only for hardly 7 to 8 years only, when you will keep it in like minus 60 or minus 80 degrees Celsius and all. But if you will go with the phenol chloroform methods, or you will go for the salting out methods, you can keep your DNA samples for more than 20 to 25 years. Your DNA life will be more like more than 20 years ago. Now, recently, when I had just visited my lab, so I had seen my lab, my DNA samples were kept very much nicely under custody in the minus 80 degrees Celsius. So, whereas if you will be going for a kit-based method, then it is hardly possible that you can store at least to the more than 8 years or 10 years. So, kit-based method is, of course, from a forensic perspective only, that you will work with your samples, and then after you will release or you will discard the samples from the data. Then the PCR reactions, of course, everybody knows about the polymerase chain reactions. So I'm not going for that particular direction, because that is a basic theoretical aspects and practical aspects though even. I'll tell you about the markers even. Okay, either a mitochondrial region markers or Y-SNPs markers. DNA sequencing we did with the 3730 capillary electrophoresis, and then after we had applied simple type of statistical analysis. But when we come for the statistical analysis pertaining to this work, especially, you have all free softwares available in the website. Okay, either it is a phyllo tree website, either it is a network tree drawing website, Either it is a MEGA, that is molecular evolutionary genetic analysis, or either is it a different type of software which is freely available. But yeah, of course, DNA sequencing is the one. Now, this is the alignments, what we see exactly. Okay. After, I mean to say that, okay, once you will get the reactions, okay, once you will get the PCR and PCR reaction, and then after the sequencing reactions, how you will be going to get the results. Okay, so prior to this, okay, before like 20 years or something, we used to work with RFLP markers, right? uh, RFLP markers, that means restriction fragment length polymorphisms, where only data was to identify the bands. Okay, based upon the bands only, we used to identify that whether which type of band it is and which type of markers or which type of nucleotide changes can it be. But here it is very much different. Here what you can observe, madam, I'm audible, right? We are continuing.
Yes, sir, you're audible.
Yeah, right, right. So now this is again a free software by edit sequence alignment. Okay, once you will get your results and all, you can target your samples. You can identify your samples like this. Now, what will happen basically is that the topmost region, okay, the topmost region is your reference sequences. The reference sequence is a similar type of database. You can go to the NCBI website, okay, and from there you can download the European model sequences. That is called as RCRS, okay, that is revised Cambridge reference sequence. It is called as the Anderson model. Why it is called as RCRS or revised Cambridge sequence? Because the first lady who was targeted for the mitochondrial DNA, she was studied in the, her sample was studied in the Cambridge University. So mitochondrial DNA was named after that organization only. So that is the RCRS it is called as. So whenever you will be working on your samples, you need to identify or you need to choose your samples based upon this particular sequence only, RCRS. So how now to identify whether is there any similarities or whether is there any dissimilarities, whether it is there or whether it is not there? Because like this, it is not possible to identify. So one way is that this software will tells you that now this is the whole data, but now when you will be using this software better, it will, this is the one, okay, where you can see the minimum differences. It is called as insertion, the green one. Green one, where it is the deletion is there, and in your black one, this is the insertion in one of the sample, right? This is the one of the area. Now see, now one-to-one samples, right? The RCRS means your reference sequence, when you will be just identifying. So all the models, okay, all the samples, how it is looking like. Now, constant to one, okay, the reference sequence is telling you that there is a G called as guanine, of course, but in all of the rest samples, there is a A, okay, there is a change of nucleotide, okay. Whereas parallelly, when you will go further to identify, there is a T in the reference sequence, but there is a C in rest of all the samples. So now till here, we can understand that these all four samples are together. Okay, there is all four samples either belongs to one population. These either four samples belong to a single family at least. These four samples belongs to a one clan, one community, to this extent only. Okay, but now when you are moving further, you can see the further differences. See, now you can spot one more difference. This is the sample number 1, that is A to G. This is the only difference in this sample. Whereas in the last sample, you can see or you can observe that, see, from T to C, there is a nucleotide change, right? This is the one difference for this particular individual sample. Now, how you can observe this? Again, see, there is a insertion. Okay, there is an insertion of T in two different samples in a two different positions as well. Now, you can highlight like this also, that I mean, software will give you an option like this also, that whatever the highlighted positions are there, that also you can observe separately. After that, we'll be moving to drawing the similarities and the dissimilarities in the form of phylogenetic tree, what we will call it as. Okay, what we will call it as a networking type of trees. So we say that networking, that means a mother computer will be there, simple to try to understand. A mother computer will be there, and from there, there are 3 or 4 or 5 different types of systems will be connected. So absolutely like that. Here you can see some of the similarities as well and some of the dissimilarities as well. So when we will say that what are the similarities or what are the dissimilarities, how to identify all this, you can go for the joining of the tree. Okay, this is one of the, you can call it as phylogenetic tree. You can call it as a nomenclature. This is a one community. Okay, this is the one community sample what we had studied specifically. Now it is looking like a simple circle, but now when we will try to understand more deeper, then it will tells you that these are the some of the few samples which are in one of the group, right? Then after, these are the samples which are there in a different group. The next, these are in different group, especially. Now, when we say in a larger type of area, okay, this one, okay. Now, how it is going to differentiate? How it is possible that one community specifically, but having a different type of nucleotide position or different type of nucleotide differences? So just go to the back side, okay, back one. Now, let's spot the differences. Where this is the one differences, I'm just showing you the A to G. And where is the other differences in the number 5th sample, that is T to C. Where is one more differences over here, whereas one more differences over here. But the similarities is there in the form of C in all the samples, right? In all the samples, there is a. The observation from G to A, there is a change. And from T to C, there is a change. But in all of the samples, it is. So all of the samples, when it will be there, it will tells you that the basics, okay, the how this tree is getting bifurcated. See, now, this is the one model. Moving further, the Y chromosomes, okay, the male specific regions, okay. So now, why we are choosing the male specific region? Either it is going for a paternal identifications, either going for ancestral identifications, either going for disease perspectives, either to identify that what is the phylogeographic nature, okay, or what is the phylogenetics nature. So now, apart from the mitochondrial DNA, the Y chromosomes can be studied in a different ways. That is, single nucleotide polymorphisms, microsatellites you can study with. You can study with the mini satellites as well. You can go with the insertions and deletion models as well. So insertion and deletion models are quite famous to identify the YAP type of studies, that is Y chromosome allopolymorphisms. It was, of course, previously they used to study, but nowadays it was being less. So this particular position of Y chromosome will be targeted, that is YQ11 position. So now the one more study, what you can go for the azoospermia factor. Azoospermia factor, that is 3 subdivisions of genes are there, that is AZFA, AZFB, and AZFC genes. This can be specially studied for the azoospermia factor with special reference to your Y chromosome phylogenetics, that in Indian subcontinent, which of the population is much affected with this particular genes, okay, or male infertility is how much stronger is there in that particular population. First, you can go for the phylogeographic nature, and then after you can come for the AZF ABC gene studies. How we will do these studies especially? So now, this is a Y chromosome consortium tree, okay, YCC. This here we are not going to identifying any new models or new type of studies. Instead, that we are just identifying or we're just taking with the help of the older type of SNPs, and with that particular SNPs, we are trying to work on our population based models or our population specific models. How is it possible to either categorize or how to categorize the individual population through this YCC tree or Y chromosome consortium trees? So now there are some published data which says that one haplogroup, that is Y chromosome A haplogroup, is mainly suitable for one population, that is Africans. Y chromosome haplogroup B, that is specially focused for the Americans. Y chromosome haplogroup C, D, and E, that is again especially quite defined for the European type of haplogroups. But now coming for the India and nearby Indian subcontinent, like India is there, Pakistan, Nepal, China, and the Asian Pacific continent, okay, Asian specific subcontinent and all. So there we can trace this particular type of haplogroups. Okay, whatever now you are seeing that M201, M52, M69, P19, M170, and this everything, these are primers. Okay, these are called as a DNA markers, Y chromosome DNA markers. When we will do to analyze these particular primers or Y chromosome markers, then how it is possible to move further? This everything is there in the Q arm, okay, short arm of the Y chromosome. So now, this is the ready-made data, what is available now. Okay, if you are working for your set of population or your set of community or your set of research area or research interest, what you can do basically is that just go to identify this particular M9, M45, and M89. So somewhere here, okay, somewhere here you will be going to identify the M9, that is K haplogroup. If M9, that means there is a complete set of 340 base pairs is there in this primer. And in this particular 68 nucleotide position, if C to G is there, then only your K haplogroup is identified. Okay, let me again tell you, okay, there is a model Y chromosome consortium which says that M9, which tells you if there is a 340 base pair length of primer, if you are working for your sample, and if in reference sequence there will be a C, but in 68 position, if G will be coming to in your samples, then that particular G samples can be separated and identified that it is a K haplogroup samples. What you will do is you will again move further for M45 or M89, like see M52, M69, and further P16, M82. So every primer is having a set of nucleotide PCR base pair sizes and a mutation size, that wherever there will be a mutation, which position there will be a mutation. So unlike the mitochondrial DNA, randomly you will get the mutations anywhere in the mitochondrial control or coding region. But in a Y chromosome haplogroups or Y chromosome region, you will be getting only these notified areas only. So rather than this, you won't find any new mutations unless and until any disease effects are there. So of course, we will be not going for a disease identification unless and until you will be sorting out your samples for azoospermic factor or infertility or any male specific type of disease or disorders. So there are some number of primers are there which says that this particular set of primers are designed. These are the universal primers, let me tell you. Okay, these are the again universal primers. That means if a person sitting in a Bangalore want to carry the research work in this particular area, then he can do. If a person sitting in a New York also, he or she will be targeting this region of primers only. But the only difference will be the populations, the models, the specificity that will be different. But the chain, I mean, the primer length will be the 534 only. In the 477 base pairs, there will be A to C mutations will be there, which tells you that that particular haplogroup is present in that particular population. So further, we had identified that our samples were identified like matching with the Sri Lanka and Pakistan as well. Okay, what we had derived is M69 in our samples. Haplogroup G is very much constrained to the India and Pakistan as well. Okay, so like Kashmir, okay, you cannot say like a one area specific for that particular aborigines or something, but the haplogroup G is majorly noted down in the Pakistan and the very much upper region of the belt. Okay, haplogroup O is majorly in our tribal populations in Indian subcontinent. Tribal populations because they are the very much ancestors. Okay, they are the one who had migrated to the Indian subcontinent at the first. Okay, then after the population, population were divided because when you will go for the history or when you will go for the genetic history, when you will go for the genomic history, there were no caste, there were no creed, there were no tribe, there were no something. But 1 lakh years or 5 lakh years, whether there was a human being or not, there were so many times the question arises. And then after the answers will come that Neanderthals who were survived 2,50,000 years ago. Okay, the Homo sapiens who had survived 1,20,000 years ago. So from there, our ancestors had migrated. Okay, and along with this, we can also identify that from where our ancestors had identified or where they had from migrated. Okay, North Asian populations who are having a haplogroup P, the M45 primer will be identified by this. The haplogroup L, that is M11, which was constrained to your Central and Western Asian regions specifically. As I told you that we always should not think only in the perspective of forensic field and criminal justice system. Of course, we are meant for that thing, but we need to collaborate our research in such a way that it should be helpful for the society as well. It will helpful for the multidisciplinary actions because we are working in a multidisciplinary models. So only forensic science that is not constrained to only a few small room. Okay, so it should be quite enlarged. It should be quite disciplined way. Okay, it should be like go beyond the limits like human population, wildlife, or pathological things or complex traits, diseases and all. So in future, what is going to be happen is that multiplexes what had came also. So in this particular type of works, now multiplex PCR set came. Multiplex PCRs, that means all these primers will be involved in a single set of multiplex PCR. And at a stretch, you can work for all these primers togetherly. And you can identify that whether there is a mutation present or not, and whether there is any insertion, deletion is present there or not. So we can futurely work. We can futurely work with the complex trace diseases. Okay, and the crime scene investigations, the biological relationships, and further different models which are going to be invent or which are going to be show a new different directions as well. Thank you all of you.